Hello, welcome to this short video that's going to be all about viral structure, and we'll also cover some aspects of viral replication as well. So it's all focused on the AQA A-level specification, looking at the 2 points there. So one which is all about structure of cells and viruses, and another one a little bit further on about how cells replicate, specifically looking purely at the viral replication process. So let's get going. So what we've got here are two little diagrams. So I've got a diagram at the bottom of sort of the structure of a viral particle, and at the top we've actually got a computer enhanced micrograph image of the coronavirus. The most important thing to remember with viruses is that they are acellular. That means they are non-living particles. They cannot carry out any functions unless they have infected a host cell and taken over the host cell. So a virus on its own is inert. It's only going to be take over the cell and carry out replication, etc., once it is inside a host cell. So they are non-living particles. They cannot carry out respiration, replication, or anything without a host cell to do it for them. They are very, very small. They're absolutely tiny. They're between 20 and 400 nanometers roughly in size. There is a bit of variation between viruses and both plant viruses or bacteriophages, which are viruses that infect bacteria, compared to viruses that infect humans or animals. So there is differences in size, but they are all very, 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 very small. So the main thing for our A-level spec is that we need to understand and be able to identify specific viral structures and structures within the virus. So let's start sort of in the middle bit, the most important part, right in the centre of my virus here, my little diagram. So this is a nucleic acid. All viruses will have a nucleic acid. Now it could be DNA, but it may also be RNA, and this is where their genetic material is. It's going to contain the genes required to reproduce more viral particles once it is inside a host cell. Remember, viral particles only replicate in a host cell. Sometimes you see different references to viruses. We might have heard of a retrovirus. So retroviruses will have RNA as their nucleic acid. And I'll explain a little bit further when we get onto a future slide what we mean about retroviruses. So surrounding that nucleic acid is a really important little structure. It's called a capsid, and it's essentially a protein coat, and it surrounds and encloses that genetic material. So it's a sort of a protective layer, really. Now, surrounding the capsid can sometimes be a second layer, and this is called a lipid envelope. Some, but not all viruses, as well as the capsid, also have this lipid envelope. So an example of a virus that does have a lipid envelope would be the human immunodeficiency virus, or HIV. So this lipid envelope, again, is another sort of protective layer, and it also aids a little bit about how it can enter the host cell in some cases. So again, we'll look at that structure a little bit further on when we look at viral replication. So along that lipid envelope on our diagram, we can see we've got these sort of stalk particles that sort of stick out. These are what we call attachment proteins. Now, if a virus has a lipid envelope present, like the HIV, what we will find is that the attachment proteins are on this outer lipid envelope. Like I said on the previous slide, not all viruses have lipid envelopes, and if they do not have a lipid envelope, what you will find is that these attachment proteins are actually sticking out and part of the capsid. They're really, really quite important. They're essential to how the virus can identify and attach to host cells and infiltrate into the host cells, so it can trigger that ability to gain entry into a host cell and allow it to be replicated within it. So it's vital sort of to viral replication and production. So it's almost the key to get in as we go. Now, this particular virus, that diagram we've got, we can see these little round dots, you sometimes see them within the capsid, has got an enzyme in it. Now, the enzyme is usually reverse transcriptase. Now, reverse transcriptase as an enzyme will be found in those viruses that are classed as retroviruses. So viruses where their genetic material, their nucleic acid, is RNA. Reverse transcriptase, and I always think of it does the reverse of transcription. So transcription normally converts a messenger DNA into a messenger RNA signal. What reverse transcriptase is, it takes the RNA and converts it back into DNA. So it's going to convert the viral RNA with the genes in it into viral DNA, inserted into a host cell genome for replication. So really, really important little enzyme there. So you sometimes find some other enzymes that are involved in basically breaking down the capsid once it enters into the host cell as well. But reverse transcriptase is a key enzyme found in those retroviruses and that are there involved as part of that conversion of the viral RNA into viral DNA for insertion into the host cell genome. So viruses, because we said at the start, non-living, they cannot replicate themselves. They require a host cell, and they require a host cell to replicate them for them. And there's some key little steps and events that are going to be carried out that allow them to go through the steps and take over a host cell's reproduction, and then to allow them to move out of that cell as well. So we're going to start at the top of this diagram. The very first stage, and the thing that's going to happen, we've got these little black blobs, these are my viruses, is that the viral particle will attach to a receptor on the surface of the host cell. So it'll use its attachment proteins either on the lipid envelope or on that capsid layer, depending on whether it has a lipid envelope or not, and it will bind to and attach to its target host cell. Now, there are two things that can happen here, and it kind of depends on whether it's got a lipid envelope or not. So viruses that have a lipid envelope can merge and enter the cells into the host cell by a form of endocytosis. In some cases, the binding of the attachment proteins on the viral particle to the host cell can also basically trigger a form of endocytosis, and they allow the viral particle entry into the cell. Now, what happens once that viral particle is part of the cell is that we're going to start removing those outer layers. We're going to have this uncoating process, and we're going to release the DNA that's surrounding them. So quite often the capsid proteins might get broken down by the cell enzymes found naturally within the cell itself, or the viral particle may have enzymes within it that are triggered to start the process of breaking down the capsid. So it can be an either or. Once inside the cell, once we've uncoated and released the DNA, what the DNA is going to do is going to viral nucleic acid, if it's an RNA, will be converted to DNA using the reverse transcriptase. Or if it was a DNA-based virus, so the nucleic acid contained within it was DNA, it's going to become part of and hopefully be inserted into the host cell genome. And we will see that that DNA will get replicated multiple times within the genome. We'll also see that it gets transcribed and allows for translation of messenger RNA to produce the proteins required to make more capsids, make more of that viral structure, more reverse transcriptase to pack into these viral particles. So we have this process whereby the viral genome is replicated, translated, and transcribed and translated, and then we have a process at step 5 of this assembly of these viral particles containing just the same structures we had before, a simple genetic piece of genetic material, so a nucleic acid, either DNA or RNA, surrounding by a capsid. And that leads up to, once we've started making these viral particles, is that they will then be released from the cell. Now, the viral release can happen in 2 different forms. It can happen in what we call a form of budding, or we can also have it through lysis. Now, in budding, what happens is the viral particles bud off the surface and take with it some of the host cell's membrane. And this is one of the ways in which viral particles can gain a lipid envelope surrounding their capsid. When it's a budding process, we call it cytopathic because it does not necessarily lead to the death of the host cell. It does allow it to, like I say, acquire its lipid envelope. And an example of cells that can do sort of viruses that carry out that budding process would be the flu virus. It buds off, taking with it some of the plasma membrane, generating its own lipid envelope, and then it can go on to infect more of your cells. Other viruses, they get released from the host cell by a process of lysis. And what we talk about then is we say that this process is cytolytic, and it can lead to cell death at the end of that for the host cell. One example of a virus that carries out that kind of replication and release from the cell is actually the virus variola major, which is the virus that leads to smallpox. So we've got two examples of how they're released. But in both cases, the virus particle is taken over that host cell. It's taken over all its replication. It's taken over its protein synthesis to become almost a viral factory, to make more and more viral particles, up until that point that if it is a virus that is released by lysis, that it can lead ultimately to the death of that host cell. I hope you find this useful. There's going to be, again, look back over this. There are some links to some other topics as you go through. So thinking about the structure of ribosomes, RNA, and even the process of that enzyme reverse transcriptase. Thank you very much for listening.